Alright, so jumping back into the M phase, which represents mitosis. So we know this is really focusing on dividing the nucleus, so nuclear division. So we're trying to divide the DNA into 2 nuclei. And when we're done, our goal is to have identical chromosomes in each of them. So our goal for mitosis is to make 2 genetically identical daughter cells. So we know we have PMAT, like we talked about before, prophase, metaphase, anaphase, telophase. And really the purpose of mitosis, this is when we are going to make more body cells. Okay, so this is when we are growing. Okay, so as you grow, you need more cells because you're not just one big blob of a cell like we've talked about. What with the problems with surface area and how that's an issue. So we need to make more cells as we get larger. Mitosis is also used for repair. So when we cut our fingers, we know we need to make more skin cells to heal that wound. And so we do that by doing mitosis. And then this process is essentially what bacteria do. So we call it, binary fission, or but essentially it's the same process of mitosis. So this is used in asexual reproduction as well. And then we go into cytokinesis, which happens when the cytoplasm divides and individual cells are created from the one original cell. Okay, so just looking a little bit more detail about what is going on here. So looking at the spindle fibers. So this is one of the things that we need to focus on a little bit when we're thinking about the cell cycle. So at the beginning of mitosis, the chromatin becomes the chromosomes. And we have centrioles that are moving to opposite poles of the cell. Okay, so remember those centrioles, those are unique to animal cells. Okay, and this is what they're going to help make the spindle fibers. Okay, just a little bit more details about the spindle fibers. They are made of proteins, specifically the protein actin. And they're made up of microtubules, and those spindle fibers are going to form between the centrioles. So like we said, the centrioles are unique to animal cells. So plant cells don't have them, but they're still going to form spindle fibers. They just use a little bit different structure to make that happen. All right, so you can see here we've got centrioles that are producing those spindle fibers, and those spindle fibers are going to come out, like we talked about, like those spider webs, shoot out and attach to the chromosomes, and then they're going to pull back. And as they shorten, they will pull those chromatids apart. All right, so this is all happening in anaphase. And we know when we think anaphase, we're going to think apart. So when we're done, after they've been pulled apart to each pole, each side of the cell will have its own set of chromosomes. So what gets a little bit complicated here is once they're pulled apart, once we pull those chromatids apart, we start calling them chromosomes again. And I know that gets a little crazy. So we have the chromosomes that we form during prophase, and then they pull apart, and they're still called chromosomes after anaphase. All right, so looking here, once again, we are going to divide the cell at cytokinesis. So we're going to develop or form a new nuclear envelope. And it's going to form around each of those new sets of chromosomes. And so when we're done, we will have 2 nuclei. and that DNA starts to break down and go back into the chromatin form. So hopefully you remember your chromatin. Okay, so remember the purpose of cytokinesis. We're going to divide or split the cytoplasm. Okay, so big part of the cytoplasm, not only the cytosol, but the organelles as well. So each of those new cells, we want it to have a mitochondria, we want it to have lysosomes. So that's this process of making sure each of those new cells has what it needs to do its job. All right, so one of the things that makes life a little bit complicated is when we go to replicate our cells aren't the best at replicating at the ends. So for some reason, there's a lot of issues like as we go through there, and as we look at DNA replication, we'll kind of dig into those issues a little bit more. But as we get to the ends, we don't do very well when we're trying to replicate them. So we had to come up with a system to deal with that. And so we have something called telomeres. And these telomeres are going to be the ends of the chromosomes, of eukaryotic chromosomes. So remember our bacteria chromosomes we talked about a little bit. They're going to be the circular chromosomes versus our eukaryotic chromosomes. Most of the time they look like an X. So you can see here these yellow pieces. Those are going to be our telomeres. Okay, so the problem is every time we were to go to replicate, we lose part of our chromosome. Like I said, it's just for some reason, our cells don't do very well replicating the ends of our chromosomes. So that's going to be a problem if you're losing pieces of DNA every single time you go to divide. So you can see here we have an enzyme that's going to be called telomerase, and that's going to add these things called telomeres. And you can see this part looks good. You're not losing anything after each division. Versus this is what normally happens. You're going to see a loss of DNA every single time. Okay, so we have these things called telomeres, and these are going to be protective end caps of a chromosome to help make sure we're not losing important genetic material. So we're still going to lose DNA, but we're going to lose DNA that doesn't really matter beyond just being a protective thing. Okay, so these are called telomeres. So this is a series of sequences. So we have TTA, GGG, and these are going to be repeated at the end of our chromosomes thousands of times. So it doesn't matter that we're losing these TTA, GGGs, because they don't really code for anything, but it is protecting us from losing the code, losing the DNA that actually does code for something. Okay, so we can lose a little bit with each division, and we're not losing anything important. So it's just a built-in protective mechanism. So these are called telomeres. Okay, so most of the time, we're going to lose some of our telomere every time we go to divide. But sometimes we have an enzyme called telomerase that can add DNA to our telomeres. Okay, so it's going to lengthen telomeric DNA by adding more of those repeated sequences to the ends. Because eventually, normally what's going to happen if we don't have telomerase, eventually we're going to run out of telomeres. And then you're going to start losing DNA that is important, that codes for things. So telomerase, unfortunately, is only in some of our cells, not all of our cells. So we don't get this extra form of protection in every cell. but it is found in cells that need to be able to divide an unlimited amount of times. So, for instance, blood cells, they have that enzyme telomerase. So they just add on more repeated subunits to the telomeres. The cells lining the intestine and skin cells. And then cancer cells also have that enzyme telomerase. All right, so we know, we've talked a little bit about apoptosis already. So this is cell aging that leads to programmed cell death. So normally we have a limited number of cell divisions, so about 80 to 90 times during a lifetime. And essentially the big reason for that is because we run out of telomeres. Okay, and so once we start losing telomeres, like we said, we start losing genetic information, and that's going to be an issue for our cells. So we have a limited number of cell divisions. But the ones that have telomerase seem to have an unlimited number of division because we can just add more repeated subunits to the telomeres. And the thought is that potentially this could lead to immortal cells. Cells that could divide forever. Because the thought is these telomeres are linked to aging. So if we could turn telomerase on in every single one of our cells, could we stop aging? All right, so that's like the thought there, and the importance of telomerase in cell division. All right, so hopefully you were able to go through. If you missed anything, don't forget, you can always pause and rewind and make sure that you are adding some of those little things, those little notes, as they give you some hints throughout here. Thanks.